This video is for chapter 11.2, which is all about muscles and movement. Okay, so first when we talk about movement, we need to talk about a skeleton. And not all organisms have a skeleton. Some, like this slug here, are just blobby. They don't have that. But of those who do, the skeleton is going to play two main roles. And one is for support, and the other one serves as an attachment point for muscles. So we can see that here, these muscles attaching to the bones of the skeleton. And here we can see on the inside of an insect, their muscles attaching to their exoskeleton. Okay, so let's talk endo and exoskeletons. So endo means inside, kind of like endocytosis, bringing big things into the cell. Okay, all vertebrates are going to have these: fish, mammals, birds, etc. And that means their skeleton is on the inside and it's made of bone, like a calcified bone material. Exoskeletons are on the outside, okay? Exo meaning outside, kind of like exocytosis, moving things outside the cell. And these guys are made of chitin, okay? So chitin is a polysaccharide, okay? And we've heard of that term before. And this is why if you eat these things like a bug, they're crunchy. Now, when muscles attach to bones, they form a lever, okay? So for all my physics friends, okay, a muscle is going to attach to one bone and then to another, and then it allows that bone to move in a lever motion, okay? For those muscles of an insect or other organisms that have an exoskeleton, they attach in such a way that gives that a lot more leverage. That joint has way more leverage. And that way they can do amazing things. They can carry up to hundreds of times their body weight, and we can't do that so much. They can jump what for us would be the equivalent of jumping like 10 meters up in the air. Again, we can't do these things. Our muscles that attach to our bone have a limit to the leverage they can provide, and things like insects can do a lot more than we can. All right, so let's take a look at how these muscles and attachment points work in a humans or something with an endoskeleton or something with an exoskeleton. So we're looking here at an insect and we're looking at a cross section through its belly or abdomen. So this would be its back. Here would be its belly, and of course its wings would be on the side here. So first thing I want to point out is the muscle. Easy to find in this endoskeleton organism. In this exoskeleton organism, this insect, it's this blue band right here. Okay, so then we can take a look at the anchor or attachment site. Okay, and this is going to be the part of the joint that stays still. It does not move. Okay, so for that, okay, I see some attachment happening here, and for this organism, this is going to happen on the bottom. Okay, I could have drawn that in either place. Okay, so it's attaching right there. And then we have the lever. So I can imagine how if this muscle was going to shorten, it would bring this bone that it's attached to upwards as well. And so that's how I know this bone is the lever. In this organism here, okay, I got to think about it. If this muscle shortens, okay, well, it would be shortening the other way. If this muscle shortens, it's going to bring this part down with it. And that's how those wings are going to flap. So for the lever part, that's going to be this up here. Okay, so it's going to bring this whole section down when this muscle shortens. Now, muscle contractions are great, okay, but they need what we call an antagonistic muscle. And together they make up an antagonistic pairs. And these are muscles that attach to the same bones, but accomplish opposite movements when they contract. So imagine your quadriceps on the front side of your leg, okay, and your hamstrings. they're going to both be involved in your knee joint. So when your quadriceps contract, they're going to pull your leg upwards, straighten your knee. When your hamstrings contract, they're going to bend your knee or flex your knee. And so because they are attached to the same bones, but they accomplish opposite movements, we call them antagonistic pairs. We've already looked at this with the diaphragm and those interior intercostals. So the interior intercostals, when they contract, they're going to make the rib cage move inward, okay, compress that chest cavity. The diaphragm, when it contracts, is going to accomplish the opposite movement. So antagonistic pairs. So when we say that muscles help to move things, okay, there has to be a joint involved where two or more bones come together. And we have a whole lot of different types of joints in the body. For the scope of this chapter, we're only going to talk about one type of joint, and it's called a synovial joint. Synovial joints join bone to bone, and they're surrounded by a capsule, like this jelly, well, not jelly, but this fluid-filled sac. Okay, and that fluid is called synovial fluid. So some examples include your elbow, we'll be talking more about that later, your knee, okay, really looking at things that are bone on bone, but surrounded by this synovial fluid. These things have some pretty restricted movement, so they're not so free like my shoulder or anything like that, okay, so the stability provided by the synovial fluid, or that joint capsule, kind of limits the range of motion of those joints. Okay, so let's talk about this elbow joint. There's three things you got to know for the elbow joint. You got to know how to draw it. You got to know how to label the parts, whether it's in your own drawing or in a diagram already given to you. And you have to know how to annotate with the functions. So we'll talk more about the functions in a little bit. Let's get to our drawing. I like to start with the bones of the elbow, and I'm going to start here with the bone of the upper arm called the humerus. We'll go back and label later. And before I draw the bones of the forearm, I'm going to leave a little space in here, okay, because there's some stuff in here that we've got to add in just a moment. So I'm going to start with the radius, and the radius is a bone that is in your forearm and connects to your thumb. The ulna kind of comes around this way behind your elbow, okay, and then it's going to be shaped like this, and eventually those two bones will form, both bones in your forearm, they're not fused together like we're looking at this angle. The radius connects to your thumb, the ulna is going to connect to your pinky, but from this angle they kind of look like they come together. They don't in real life. Now, anywhere where this bone and either of these bones could rub together, that could create a lot of friction and pain. So the ends of these bones are covered in a thin layer of cartilage. So let's see if I can get this right here. Okay, so there's a layer of cartilage there. And we've got a layer of cartilage covering both the ulna and the radius. Okay, so it's important to note there that cartilage is actually connected to the ends of the bones. Okay, then this is all surrounded by a joint capsule. Okay, so the entire joint is going to be surrounded by a joint capsule, and that's going to be filled with the synovial fluid. Okay, so the joint capsule kind of joins the joint together, and then it's going to fill with fluid. Now let's start in with your muscles. Both of the muscles we're going to use in the human elbow joint attach to the humerus, but the attachment point is way up here, out of the scope of our drawing. So I'm just going to draw in the actual kind of like muscles themselves. So on this side of the elbow joint, we have the biceps muscle. Okay, and so the biceps muscle kind of sits in here, you know where that is, okay? And it attaches via a tendon to your radius. So something about like that. We can see how if this bicep were to contract or shorten, it would bring this part of the arm up as well. So our next muscle is going to be on the back side of the arm. Okay, and that's called the triceps muscle. And the triceps muscle is going to connect to the ulna. So if this muscle shortens, it's going to bring the bone backwards. So this is responsible for straightening your arm. Those are our antagonistic pairs. All right, let's do some labeling. So we just finished drawing the triceps muscle. Okay, and this would be the triceps tendon. Tendons connect muscle to bones. Over here, this bone that we're connecting to is called the humerus. Okay, and here we have the biceps muscle. The biceps is going to connect the humerus to the radius. And it does so via the biceps tendon. Okay, down here again we have the ulna, and that connects to the triceps. And then we need to start labeling parts of the joint. All right, first thing I'm going to try to do is label this cartilage. Okay, so I only need to label it once. Cartilage. It'd be helpful if I labeled it correctly though. Okay, I don't need to label it on both of the bones. Okay, then we have this joint capsule. And the fluid that fills it up, called the synovial fluid. Okay, so if I were going to color in the synovial fluid, okay, that's all of this stuff in here, inside of the joint capsule. So it's kind of filling up all of this space. Okay, now you should take a moment to go through and annotate the parts with their functions. So cartilage, again, is going to reduce friction. It can also absorb any compression, okay, so that your bones don't absorb all that force and shatter. The synovial fluid is going to lubricate that joint, reduce friction furthermore, and provide nutrients to the cartilage cells. The joint capsule surrounds the joint, so it's going to kind of hold in that fluid, but it also is one of those things that kind of helps unite those connecting bones. Tendons attach the muscle to the bone. Ligaments we did not put in our drawing. So we did not draw in the ligaments of the elbow. You don't need to, but somewhere out in your margin I would write what ligaments do, just so we don't get them confused with tendons. Okay, we've got the biceps muscle. That's going to cause the arm to flex. The tricep muscle, when it contracts, causes the arm to extend or straighten out. We've got the humerus, that's the anchor point for the biceps and triceps. The radius and the ulna, which are our levers for those two muscles of that antagonistic pair. Okay, now I bet your wonderful biology teacher has continued to show you animal cells that look like this. And that is a typical animal cell. Okay, but that typical animal cell is undifferentiated. It doesn't do anything. This is just some kind of picture that someone made up. This is also an animal cell. Okay, it's a muscle cell, but it looks entirely different. So even though this muscle cell has all the same parts as this typical animal cell, they look different, and we have some special names for some of them. Okay, so let's go through those quickly. So a cell membrane that is part of a muscle cell is called not a cell membrane, but a sarcolemma. The fluid-filled portion isn't called the cytoplasm, it's called the sarcoplasm. And the endoplasmic reticulum is called the sarcoplasmic reticulum. So sarco, sarco, sarco. So muscle cells have some pretty unique adaptations to try to help them do their job. And one of the things that they have is a whole crap load of mitochondria, as seen here. So we can see the cristae, okay, and those cristae are indicators that all of these little blobbies are mitochondria. And this is because muscles need a lot of ATP. In order to get a lot of ATP, they got to be doing a lot of cell respiration, and for that, they're going to need a lot of mitochondria. They're also going to need a lot of glycogen. So we've talked about glycogen before. Okay, glycogen is a polysaccharide that's made out of, could be thousands of monomers of glucose. And we store this in a couple spots in our body. The liver is one, and the muscles are another. So when muscles need glucose for all that cell respiration, they can take this glycogen and hydrolyze it or break it down into those individual glucose monomers. As expected, muscles are going to have large amounts of ATP, okay? That's necessary for contractions. We'll talk more about that later, but for contractions to take place, we need a lot of ATP. They're also going to have multiple nuclei. So when I'm looking at a picture of a muscle cell, okay, a single cell is going to have many multiple nuclei. Okay, that really originates from how muscle cells are formed. They actually come from the fusion of precursor cells. And one of the reasons that they fuse is to allow for more continuous work and contractions. So they end up with these multiple nuclei. Okay, so let's talk about cell respiration for just a minute, okay? I know that the goal of cell respiration is to take the energy found in glucose and make ATP. Some of that energy also is inadvertently transferred into heat. Throughout that process, I'm going to need a couple of things. I'm going to need oxygen, okay? And oxygen is carried to the muscles via hemoglobin. So hemoglobin can carry 1, 2, 3, 4 oxygens, and it flows through the bloodstream. Muscles contain another oxygen-carrying molecule called myoglobin. So this prefix myo, we're going to see this a lot, okay? It refers to muscle. These are only found in muscle cells. Now, myoglobin can only have one oxygen, okay, and it really has a high affinity for oxygen. So it's going to hold on to its oxygen until the last possible moment. So when oxygen levels are really low, okay, myoglobin still has that oxygen. It can provide a last resort of oxygen storage. And muscle cells are also going to contain many contractile units called myofibrils. There's that prefix myo again. So here's an entire muscle cell, okay? And we can see within that single muscle cell, we have lots of these guys. These are called myofibrils, and they're responsible for the actual contractions. Okay, so let's see what we're looking at here. I'm going to start with the easy ones. We know what this is, okay? This is a bone. See, this class isn't so hard. A bone. Okay, and then we know what we're looking at here is a muscle. So this entire thing is a muscle. And what connects a muscle to a bone is called a tendon. Okay, and then within a muscle, you can see that I've got several repeating units. Okay, we'll talk more about that unit in a little bit, but if I kind of dissect those, I can see this portion on the outside here, and that is a blood vessel. Okay, muscles need blood, they need oxygen, nutrients, etc. Okay, and this part here running along the course is a nerve. Okay. All right, now within this muscle fiber, okay, I can see several little small tubey things of their own. So I see one here, 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 etc. One of these bundles is an actual muscle cell. And you may hear muscle cell referred to as a muscle fiber. It's the same thing. They're synonymous. I can also see a different view of a muscle cell. So here I'm looking at the end of one. Okay, if I pull one out, it looks like this. This is all one muscle cell. So I'm also going to label that down here as well. So it's just another view of one, just to show you that this whole thing is one cell. Okay, so within that cell, I've got this pink covering, and that's the cell membrane, but we don't call it the cell membrane. We call it the sarcolemma. Okay, and on that sarcolemma, I should be able to see multiple nuclei. So I see one here, 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 here. So I'm going to label it there. This is the nucleus, one of them. Okay, just remind myself that it's plural. I'm just going to leave that as nuclei. And then I've got this netting type thing over it. This is a specialized endoplasmic reticulum, okay, called the sarcoplasmic reticulum. Okay, now within each muscle cell, okay, so again, this is all one cell. I can see it has repeating little fibers, okay, and these are called myofibrils. Okay, now these are like those little Russian dolls that you take apart and there's one inside. Take another part, it's inside. It's like never ends. Okay, so our muscle is made up of many bands here. Each one is made up of a muscle cell. Each muscle cell is made up of a myofibril, and each myofibril you can clearly see has two parts. So these are strands of proteins, and those strands of proteins are called actin and myosin. So actin is going to be the skinnier one, and then we have myosin, the fatter one. And this is the basic arrangement of your muscles. Okay, so just to recap, okay, inside of each muscle cell are parallel tubes called myofibrils. A myofibril is made up of myosin and actin. Okay, and myosin and actin even have their own repeating units called sarcomeres. So a sarcomere is going to be a repeating unit of an actin and myosin like overlay here. Okay, now a sarcomere is going to extend from what we call a Z line to another Z line. Okay, so this together makes up one sarcomere. Okay, so let's label this guy. Okay, here we have in the middle this myosin, and this skinnier molecule is actin. So I'm going to leave it up to you to kind of write down how you can tell the difference between them. Okay, so you can look at your picture and figure that out. And in terms of how do muscles actually contract, well, we'll get into more of this later. But what's going to happen is myosin is going to stay still. Okay, it's kind of stuck here in the middle. And what's going to happen is this actin is going to slide on closer to the middle of myosin, bringing these Z lines closer together, making the whole thing shorter, and that's what happens during a contraction. We're making the muscle shorter. Okay, so here's what I was just talking about here. Here's what a relaxed muscle looks like, okay? When a muscle is contracted, what's happened is that this actin has been pulled by the myosin closer towards the center. So it literally slides on over and over this way. And notice how these Z lines have gotten shorter. Okay, there's less of a distance between them. Okay, so at this point you should be able to do this one on your own, right? You should be able to identify which one is contracted and which one is relaxed and why. Okay, so one of the things that we should be able to do at this point is to draw a relaxed and a contracted sarcomere. So let's split this in half. Let's do a relaxed sarcomere on this side and a contracted one on this side. Okay, easiest way to do this is to start out with the Z lines, okay, and of course to label that. Okay, then I'm going to start out with my actin. So actin is pretty skinny, especially compared to myosin. So here's my actin. And myosin is going to be here in the middle, and it's going to be thicker than the actin. And that needs to be in your drawing, okay? It has to be shown as in the middle and thicker than that actin. Okay, so if this is what a relaxed sarcomere looks like, and of course I'd want to label that sarcomere, okay, then you should be able to draw a contracted sarcomere on your own. Okay, so how does this all work? How does the myosin cause the actin to slide over it, to come closer to the middle? Well, that's through something called the sliding filament theory, okay? And what's actually doing the sliding is the actin, okay? So let's think about how that works. In order to get from here to here, I'm going to need this myosin head to get activated, and it does that by splitting ATP. Okay, so it's going to hydrolyze this ATP. That's going to release energy. Okay, and it's going to cause this myosin head to become activated. Okay, so this myosin head is then activated. That's step one, happening right here. Okay, well, how do we get to step two? Okay, so how do we go from this to this? Well, the first thing that's going to happen is this myosin head that we had beforehand is going to attach to a binding site on actin. So this yellow thing is actin, this big fat thing here is myosin, okay? And this activated head is going to attach to a binding site on actin. And this is what we call a cross bridge. Okay, so a cross bridge is literally when we have a connection between myosin and actin. We don't have that over here. Look, they're not connected. When this happens, this inorganic phosphate group gets released. Okay, so it was hanging out here after we split ATP. This goes away, and now I just have that ADP there. In step 3, so getting from here to here, okay, that ADP is going to get released, okay, so it drops off. And when it does that, it causes this myosin head to bend this way, okay? So it literally kind of throws this actin this way. You can tell how if this were to bend this way and it's attached to this, it's going to cause this to be thrown or slid this way. So that's what we're seeing here. When that ADP is released, it causes it to bend this way. And if this is bending towards the center, it's going to pull the actin towards the center. So on this little myosin head here, okay, if this myosin head bends this way, it's going to cause this whole actin molecule to slide this way. And that's how we get this sliding of the actin molecule all together towards the center. Okay, so causes the actin to move towards the center of the sarcomere. Okay, so this always reminds me of rowers because this myosin head kind of looks like an oar. So same thing here. When you're making what's called a power stroke, your paddle is in the water, right? Just like when myosin is making its power stroke, it needs to be attached to myosin. Well, to make the next power stroke, you got to pull your oar out of the water. For myosin to make its next power stroke, it needs to detach from the actin. Okay, and in order to do that, I'm going to need ATP. So the ATP allows the myosin head to detach from this actin. We don't have a cross bridge anymore, so no more cross bridge. That's gone. Okay, we've broken that. Okay, and now we're going to be ready for the next power stroke. So all of this is going to continue, okay? Okay, continue attaching, bending, and detaching as long as we have two things available. We need ATP and we need calcium. We'll talk more about calcium in just a minute, but you should be able at this point to explain why ATP is needed for a muscle contraction. Okay, so it's added in this step. You should be able to think about that and do that one on your own. Okay, so I want to talk about calcium, but before we can talk about calcium, we really need to zoom in on actin. So this entire thing is what we call an actin filament, okay? So actin filament is really this orangey stuff, okay? And these little spots here, those little holes, okay? Those are the binding sites for myosin. Now, you may notice this annoying string here covering all of the binding sites. Okay, that annoying string is what we call tropomyosin. So this band is called tropomyosin. Okay, and this band, again, covers up all of those myosin binding sites. Attached to certain points on the tropomyosin, and you may have to point this out kind of on your spot here on your paper, are these little things called troponin. Okay, so tropomyosin, the string covering the binding sites, and troponin, okay, are both proteins. So is actin, so three totally different proteins. Okay, and so here's kind of their arrangement. Actin has all these binding sites. They're covered with a protein called tropomyosin. That tropomyosin has another protein sitting on top of it in certain spots, okay, and that's called troponin. Now, troponin is going to bind to calcium. Calcium, when it binds to troponin, causes that tropomyosin to move out of the way so that it can expose those myosin binding sites. Okay, so those myosin binding sites are going to be open or clear or available whenever calcium is bound to troponin. So in comes this sarcoplasmic reticulum. Okay, so the job of the sarcoplasmic reticulum is to regulate the calcium ion concentration. We need calcium ions to expose those myosin binding sites to move that tropomyosin out of the way. And that's the job of this sarcoplasmic reticulum, okay, to regulate that. So if we want a contraction, we need calcium to remove that naughty, in the way tropomyosin. If we don't want a contraction, we need the sarcoplasmic reticulum to make sure there is no calcium so that those myosin binding sites are blocked. Okay, so just to put this into words one more time. Tropomyosin is the band, the protein filament, that covers the myosin binding sites that are on actin. On that tropomyosin band are other proteins called a troponin. When calcium binds with troponin, it moves the tropomyosin out of the way to reveal the myosin binding site so that we can get ready for a contraction. Okay, so let's take a look at this from start to finish. So for a muscle to contract, it has to get a signal from a motor neuron, and we've covered this before. Okay, so that motor neuron is going to be filled with neurotransmitters called acetylcholine. So when the action potential reaches the end of this motor neuron, it's going to be flooded with calcium ions. The calcium ions force those vesicles full of neurotransmitters to release their contents into the synaptic cleft, and their contents just so happen to be acetylcholine. The acetylcholine is going to bind to receptors that are on the sarcolemma, so on the cell membrane of that muscle cell. That causes a sodium ion channel to open, okay, and move into the muscle cell. Okay, so that acetylcholine binding causes sodium to enter the cell, okay, and then that in turn triggers the sarcoplasmic reticulum to release its calcium. Once the calcium ions are in there, those calcium ions are going to bind to troponin and cause the tropomyosin to move out of the way so it reveals those myosin binding sites. Then we're into the sliding filament theory. Once those heads are exposed, the myosin heads attach to the binding sites, forming a cross bridge. That's definitely something you need to put in your mental tool bank. The myosin head is going to flex towards the center of the sarcomere, so something like this. And then I'm going to need ATP to bind to the myosin to get it to detach. Once it's detached, it's going to bind to another binding site further down, and it's going to repeat that binding, flexing, and detaching cycle until this actin has slid all the way towards the center, shortening the sarcomere and making the muscle contract. All right, so we often think of muscle contractions when we're living, but how about after we're dead? Well, then we get this situation known as rigor mortis. So rigor meaning stiff, mortis meaning death, so a stiffness after death. And this can last anywhere from 1 to 4 days after death. So let's think about it. When we die, okay, we're not generating any more ATP. So without ATP, our muscles, okay, these myosin heads can't detach. They can't detach from those actin binding sites. So if they can't detach, then they're stuck in a state of contraction and they can't move. Okay, so whatever position the body is, it's going to be stuck in that state or contracted in that state because these actin heads or myosin heads are stuck in that position on the actin. Now, eventually after death, okay, this tissue will start to decompose and break down, so we kind of become pliable again, but that's how we get rigor mortis. And that'll do it for chapter 11.2 on movement.